So today we're going to continue talking about T cell activation. And as I've sort of mentioned, this is the overview of this process that we're thinking about, where we start with a naive T cell. That naive T cell interacts with MHC plus peptide on an infected cell that's presenting antigen, and then sort of goes through this process of becoming activated. And we saw the signaling that happens downstream of the T cell receptor last time. If you recall, I told you last time as well that in reality, there are multiple signals that the T cell needs to receive, sometimes referred to as signal 1, signal 2. Sometimes people call this one signal 3, but that's not said as often. And we're going to see some details regarding those other signals today. This one that I'm kind of saying, I've heard people call it signal 3, but it's not, not always, is really a signal through some kind of cytokine. And in fact, there are many different cytokines that can be doing many different things in this situation. I'm going to talk a little bit about some of the cytokine actions, and then I'm going to talk more about signal 2. There are a few different cytokines that one could think about in this situation. But the cytokine that we're going to think about the most right now is a cytokine called IL-2. And you can see IL-2 being produced sort of at the very beginning of this process of our T cell being activated. You can also remember that on Friday, when we talked about signal transduction in activating our T cell, the goal on Friday was to activate that T cell to make IL-2, to transcribe the IL-2 gene. And so what you can notice is that the T cell is itself making this cytokine IL-2. And so the first thing we need to think about is sort of what's going on with IL-2. When IL-2 was first described, it actually didn't have the name IL-2. When IL-2 was first described, it was called TCGF, which stood for T cell growth factor, which should help you understand what IL-2 does. Because the big thing that IL-2 will do, and you can see a T cell making IL-2, and then that IL-2 actually stimulating the cell's own IL-2 receptor, is that pushes that T cell into the cell cycle and allows that cell to undergo a bunch of cell divisions. So that's going to allow both this proliferation of that cell. It's going to give us division on that cell, as well as allowing that cell to survive. And you can see this sort of all happening here as well. Our cell is going to be making IL-2. That IL-2 is going to feed back and bind to the IL-2 receptor that I will say more about. And one of the big things that's going to happen as a result of this is we're going to get expansion of that cell. So we're going to make more copies of that cell. One quick thing to think about with that expansion or that cell division that's happening because of IL-2. This slide mentions that this is T cells in the periphery. So specifically, where is this happening anatomically? Yeah, David. Lymph nodes. So we're seeing this massive cell division in the lymph node. What happens in a location where you might have massive cell division? What do you think, Jonah? It's going to swell. So if you think about situations where you get swollen lymph nodes, the reason why you're getting a swollen lymph node is because of this proliferation. It's getting crowded in there, and you're actually starting to get those lymph nodes swollen. So you can sort of see all of these pieces coming together. So IL-2, as you can see here, is going to signal through the IL-2 receptor. And that's going to lead to things like that cell dividing. It's also going to help that cell start its differentiation process. We'll see more about the differentiation process. But basically, the idea with the differentiation process is the cell is going to gain functions it did not previously have. The naive T cell doesn't really have any function. It doesn't have the ability to be a killer cell or a helper cell until after it has seen antigen and after it has differentiated. So the IL-2 receptor is shown on this slide. And what you can see about the IL-2 receptor, which is shown over here on the left, is that it has 3 different protein chains that make it up. One of them is called the IL-2 receptor alpha chain, one of them is the IL-2 receptor beta chain, and one of them is called the gamma chain. What you can also notice is that this gamma chain that's shown in light blue is also shared as a part of the receptor for some other cytokines. So there are other cytokines that use this gamma chain paired with different partner proteins. In the case of IL-15, it's actually even the same beta chain and just a different alpha chain. With some other ones, it's basically just one other specific chain along with gamma. All of the cytokines that are shown here are known as cytokines that are part of the common gamma chain family. This also is missing one, which is IL-21, as that's a member of the common gamma chain family. And a lot of these cytokines are cytokines that are particularly important in lymphocyte function. They're particularly important in a lot of cases, but realize that sort of a lot of the big lymphocyte functions are controlled by common gamma chain cytokines. You've actually already met one earlier this semester, which was IL-7, which was involved in helping the developing B cells and T cells as when they're sort of stem cells to develop. So IL-7 is a really nice survival cytokine for developing lymphocytes. We're going to think a lot about, we're going to be thinking right now about IL-2 and IL-2's receptor. One of the reasons why I like to introduce you to the entire common gamma chain family of cytokines here is that there is a very famous immunodeficiency disease where patients have a mutation in the common gamma chain. And so they don't make this light blue protein. As a result, they don't have a receptor for IL-2. But they also don't have a receptor for IL-15. And they don't have a receptor for IL-7. And they don't have a receptor for IL-9. And they don't have a receptor for IL-4. And they don't have a receptor for IL-21. That's not shown here. So basically, their ability to respond to all of these cytokines is gone. And patients who have that mutation have something called SCID, or severe combined immunodeficiency, which basically means they have no B cells, they have no T cells, they have no NK cells, basically no adaptive immune response because of this loss of response to all of these cytokines. The common gamma chain gene is found on the X chromosome, and so this form of SCID is X-linked and is found much more commonly in boys. If you've ever seen kind of like the bubble boys who have to be protected from a lot of microbes, they are X-linked SCID patients. Fortunately, we can treat these folks really easily now by giving them bone marrow so they can redevelop an immune system that has the common gamma chain. And so here you can see that IL-2 receptor in more detail, including the common gamma chain as well as the other 2 chains. And what I want you to notice here is that when the cell has the gamma chain as well as beta It can kind of bind IL-2. It has sort of this intermediate affinity for IL-2. So it can bind IL-2 okay. Once the alpha chain is also present, now this receptor can bind IL-2 way, way, way better. And so sometimes we see just this intermediate affinity receptor with the gamma chain and beta. Sometimes we'll see the alpha chain as well that will be high affinity. I will also point out, because this will come up more in the next class, the IL-2 receptor alpha is also known as CD25. And CD25, you could imagine if a cell has CD25 on its surface, it's going to be, we sometimes refer to it as an IL-2 sink. But you could also think of it as being like an IL-2 hog, because it's going to bind IL-2 really strongly and like steal all the IL-2 so maybe nobody else can get it. And so that's one thing to just be aware of. CD25, like I said, will show up again later. And so you can see the same idea here. When our cell is completely naive, that cell will have the gamma chain and the beta chain and have sort of this medium affinity IL-2 receptor. As that cell gets activated, as that cell starts to make IL-2, it's also going to make the alpha chain and be able to really bind to that IL-2 well and get a really nice signal from IL-2. You can see this process here as well. So here you can see, here's our naive T cell. It has not yet been activated. We haven't started the signaling we did last time. This cell does not yet have its function. And it has just that intermediate IL-2 receptor on its surface. Once we start giving this cell signal, the cell is going to start making IL-2 like we saw last time. It's also going to start making a lot of other things, including that alpha chain. So now we're going to get the high affinity receptor, and we're going to make this cytokine. And now this cell can get boatloads of IL-2 and start dividing and moving this way. Yeah, David.
Would the production of the alpha chain after getting activated prevent the IL-2 that's being produced from activating T cells, or I guess from causing proliferation of T cells that aren't active?
So that's a great question. This might kind of make this T cell be a bit of an IL-2 hog, as I mentioned. But it's not going to be super specific. And so really the reason why we're getting this division is because we've gotten the signal 1, signal 2 that we're going to talk about, and the IL-2. In theory, if you got just IL-2, you wouldn't do as much dividing, but that's a whole other argument for a whole other time. One other thing that's sort of related to what David just asked is that IL-2 is a sort of unique cytokine. The cell can make IL-2 and sort of feed itself. That is called an autocrine loop. But the IL-2 could also be made by one cell and sort of feed neighboring cells. It's not going to go into the bloodstream and like feed every T cell in the whole body. But if we're in the lymph node, it's possible that an IL-2 molecule that David makes might help Brooke. And one that Brooke made might help David. When that's called paracrine signaling. And so we do see IL-2 working in both autocrine and paracrine. Almost no other cytokine works in autocrine. IL-2 is really like the big autocrine signaler. In thinking about cytokines, we can also think about the signal transduction pathway that happens with cytokines. So we saw a bunch of kinases and transcription factors last time, and there are some specific kinases and transcription factors that are involved in cytokine signaling. So here you can see a cytokine receptor with 2 subunits, a blue one and a purple one. And what you can notice is that those cytokine receptor subunits interact with another protein. So they are closely associated with this other protein. And you can see this other protein's name is JAK. So the K in JAK stands for kinase. So the JAKs are kinases. They're normally apart from each other, but you can see when the cytokine molecule binds to the cytokine receptor, that brings the JAKs close to each other, and they can phosphorylate each other. So this JAK will the green JAK will phosphorylate the blue one will phosphorylate the green one. There are 4 different JAKs. So there's actually JAK 1, 2, 3, and the other one is not called JAK 4 because why would it be useful? The reason why different cytokines will use different JAK combinations. One of the reasons why it's important to talk about all of this is that one of the big drugs classes that people are now using to block some autoimmune diseases are JAK inhibitors. The JAK inhibitors have been approved for less than 5 years and are some of the new autoimmune disease drugs that are out there, though they seem to have pretty bad side effects. But they are kind of the new hot class because, of course, if you could inhibit cytokine responses, then perhaps you could inhibit some kind of immune response or something like that to stop having your excess immune response in autoimmunity. So what you can also notice is that the JAKs clearly have to have 2 parts. They clearly have to have a part that is a kinase and a part that can get phosphorylated. Like they've got to have a tyrosine somewhere to get phosphorylated, and they have a kinase part. So you can think of them as having 2 parts, 2 faces. They were given the name, if you read in a textbook, you will find that the reason why these are called JAKs is that JAK stands for Janus kinase. Janus is a Roman god who had 2 faces. And so the idea is that these are kinases with 2 faces, the one that gets phosphorylated and the one that does the phosphorylating. And so that sounds all very fancy and nice. When I talk to like super old immunologists who were actually in the field at this time, they've told me that that's a lie. That in fact, they first found these in some autoimmune patients and some immunodeficient patients, and they were like, "Oh, we learned a really important thing that is important for T cell development," and then they're like, "Oh, actually, no, we just discovered just another kinase." And so really old people tell me that JAK stands for just another kinase instead of Janus kinase, but everywhere you look, you will always see Janus kinase as the definition. Once the JAKs are phosphorylated, which you can see here with their lovely pink dots, that provides a binding site for the next protein in this signaling pathway. And the next proteins in this pathway are called STATs. And so you can see that the STATs will bind to the JAKs, and the STATs can also get phosphorylated. There are 6 STATs 1 through 6. Again, there's some different pairings of different JAKs and different STATs that are important in other situations. When the STATs are phosphorylated, they form a dimer, which you can see here. But then they do something important, which is that those STATs, once they are phosphorylated and make a dimer, go directly into the nucleus and are the transcription factor. STAT stands for signal transducer, because see, it's transducing a signal, and activator of transcription, because it's also acting as a transcription factor. And so the signaling pathway that we generally talk about for any cytokine is known as the JAK-STAT pathway. And hopefully, having now seen the pathway, you can understand why it's called the JAK-STAT pathway, because it has 2 parts, JAKs and STATs. And hopefully, if you can remember the JAK-STAT pathway, you can remember all the steps of the JAK-STAT pathway, because there's 2 steps, the JAK step and the STAT step. Yes, Brenna. Mhm.
So the first part was the proximal signaling, and then the second step is the cytokine signaling.
So the first step is really that's a great question. The first step is what's shown here as signal 1. So I would say that the proximal part of signal 1 are the places before we get branches. And then we got some branches. So all of last class was about signal 1, which is the signal that's coming through the T cell receptor. We just talked about cytokine signaling that can happen to help that T cell get turned on. That might be autocrine signaling with that cytokine, like IL-2, autocrine meaning that the cell is feeding itself the cytokine. That could also be some paracrine signaling, where the antigen presenting cell is giving the cell cytokines, though we're really going to see the best examples of that on Wednesday. So does that make sense, Brenna? Okay. But the other thing that we have to think about is super, super, super important. And it is this additional signal known as signal 2. And I will tell you that I think that there are a lot of questions that sometimes people can have, and signal 2 answers them. So you really want to think a lot about signal 2. And so the sort of take-home message that you will see on subsequent slides is that in order for a T cell to be activated, that T cell, and specifically in order for a naive T cell to be activated, so the T cell has never seen signal before, it's getting turned on for the first time, that T cell requires signal 1 and signal 2. It must have signal 1 and signal 2. If that T cell does not get signal 2, we don't activate the T cell. And so signal 2 is super, super, super important. We've already talked about ways that the antigen presenting cell or the infected cell makes signal 1. That's just MHC presentation, right? It does all the steps of MHC presentation. But what you might realize from what I just told you is that it's pretty important that that cell has a way to provide signal 2 as well, not just signal 1. And so we need to think a little bit about what's up with signal 2. You can see signal 2 in both of these images. And if you look at your image from your textbook here, you'll see that on our naive T cell, the protein that we're thinking about is a protein called CD28 that is receiving the signal. And CD28 is binding to a protein called B7 that is on our antigen presenting cell. So you can see B7 and MHC are on the same cell, T cell receptor and CD28 are on the same cell. There are different names for B7. I will tell you more about some of those other names in a little bit. But what I want us to think about first of all is sort of, okay, we learned earlier how exactly we get this MHC plus peptide on the surface of the cell. How did we get the B7? What's up with the B7? And you've actually already seen the details of how you get the B7. You just didn't know you cared about it. This is part of dendritic cell activation. One of the most important proteins that is made by a dendritic cell when that dendritic cell gets activated is B7. So when we are activating our antigen presenting cell, especially a dendritic cell, that cell is going to start making B7. You also saw B7 at one point earlier in this semester when you were looking at the Medzitov and Beutler papers, particularly it was in the Medzitov paper, that if you look back, B7, it's also known as either CD80 or CD86. I can't remember what they labeled in the Medzitov paper. This is the paper with the Nobel Prize and the debating. You actually saw they measured B7 in that paper and B7 production in that paper. Because B7, this second signal provider, is key for turning on T cells. And the reason why a dendritic cell or any other cell type makes B7, which is shown here as this yellow rectangle of a co-stimulatory molecule, is because that cell is responding to some kind of PRR stimulation. So when a cell is responding to PRR stimulation, it makes a bunch of cytokines that we've talked about, but it also makes B7. And so we can think about this a little bit for a second. We'll see this coming up more as we sort of keep discussing this. But this is actually a key aspect of activating T cells. As I told you previously, if you don't have signal 2, if you don't have B7 to interact with CD28 at the same time that you're getting your T cell receptor signaling, then the T cell doesn't come on. You got to have both of these things. I'm going to sort of explain this in the words of a theory that someone came up with that was a little bit controversial. I think that the way I'm explaining it is not controversial. It's all pretty legit at this point. Though I'm going to use a word that some immunologists really hate, and part of me is going to feel like I'm going to get struck by lightning by using it, but that's okay. We're going to make it work anyway. Which is that the idea here is that when the cell gets a PRR signal, that's a signal to the cell there's a microbe around. There's something bad around. Sometimes people talk about this as a danger signal. There's danger. There's a microbe. There's something that's not supposed to be here around. So as the dendritic cell, I'm going to present whatever peptides I have, but I'm also going to tell you there was some danger around. And so you T cell should probably take this seriously. And the T cell is only going to be activated if that T cell sees an antigen that it recognizes in the context of sort of danger or with this additional danger signal. This dendritic cell is saying, I saw a microbe, it stimulated my PRR, and these are proteins I have in me. You probably want to be careful of them. So does that kind of idea make sense? So let's think about sort of the other piece. There's another piece that's related to this. What I have told you a bunch of times is T cell can only be activated if it sees signal 1 and signal 2. Can you imagine a situation where a T cell might see signal 1 but not signal 2? What is a situation where a T cell could see its peptide antigen presented on MHC without signal 2 around? Yeah, Brooke. Self, exactly. Remember how we talked about the fact that all of our cells are presenting, particularly class 1, self 24/7. So we're going to be showing those self antigens all the time. And we talked about the fact that with T cells coming out of the thymus, all of our T cells are a little self-reactive. So you're going to be showing self-antigen all the time. You're going to be having T cells that could bind to that self-antigen all the time. if we're in a situation, like Brooke correctly points out, where we have a T cell that sees self-antigen and there's no B7 around. This is just, you got a T cell that responds to a brain antigen and it goes to the brain. But it's just a random Tuesday. There's nothing actually wrong there. What do you want? What hopefully should happen? What do we want to have happen to us as the organism? Or what do we want going on with that T cell? Yeah, Brenna. We might want to recycle the T cell, okay? What other kind of things can we think of? That's one way of kind of thinking about it, yes. Yeah, David. We might want that T cell to be non-reactive. First of all, we don't want that T cell to be reactive, right? But when you say you want it to be non-reactive, what do you mean? So it's more than that. So David's thinking about you want to not have a signal. But it turns out you actually want to say, T cell, you shouldn't, you know what, you might be self-reactive. We're going to turn you off. So you actually want to turn that T cell off. If it's going around getting signal 1 in the body without getting signal 2, that means it's kind of actually doing self-reactive things in the periphery. That's not a good idea. So if a T cell gets signal 1 and signal 2, then we activate that T cell. But if a T cell sees signal 1 without signal 2, then we get anergy. And so our T cell undergoes anergy. And you saw the process of T cells or B cells undergoing anergy earlier in the semester. And so if you want to activate a T cell, you actually must give that T cell signal 1 and signal 2. And if you give it only signal 1, you are going to actively turn that T cell off. Because this T cell is risky. It's binding stuff in the periphery when there's not danger around. Yeah, David.
Are there any microbes that inhibit the production of B7?
Yes. I can think of one off the top of my head. So yes, there are microbes that will inhibit B7 production. Yes, Juliana.
When you were saying the T cell will see the self-antigen B7 without B7,
Mm-hmm.
yeah, I was just wondering if you can like repeat like if it sees self-antigen, like why wouldn't it have B7?
So if we have a situation like what you see here on the right, we're going to have our T cell presenting self-antigen on MHC just because it's all day. It's like, this is the protein I got. Here you go, T cells. Am I okay? This is the protein I got. Here you go, T cells. I'm okay. If it doesn't have any pathogen stuff, it's going to be presenting self. So it's presenting self all day long, right? If it did not see a pathogen, nothing is going to trigger PRRs. Nothing's going to sort of trigger and say danger. And so that T cell is not going to make B7, also known as either CD80 or CD86, because there's B7-1 and B7-2. And so in that case, there's no danger. This must be a self-antigen because there's no danger around. And so we're going to say, T cell, you should be turned off because we don't want you responding to self-antigen. Yep, Brooke.
So it's always presenting self, but if the T cell binds to the self, that's going to turn it off?
Yeah, in the periphery. If the T cell binds to signal 1 without signal 2, then it gets turned off. Yep, Jonah.
Is it different from B cell energy where we turn it off and then said, all right, don't turn it back on in almost like emergency situations?
It's exactly the same. So it's the idea is just like with B cell anergy, if we're in a super emergency situation, maybe we need that self-reactive T cell to be cross-reactive against something else. And so it can get turned back on in sort of that emergency situation. But in a perfect world, we're keeping it off all the time. Josh.
So, if we're talking about class 1,
Mm-hmm.
nucleus cells presenting
Yep.
some antigen, can those nucleus cells also produce B7 or is it just dendritic cells?
Oh, that's a good question. Dendritic cells are by far the best at it. Though for our purposes, I will say that all nucleated cells can produce it a little bit. I will also say that there is a whole extra process that I skipped that is involved here that gets at what you're saying. But so I'll say for our purposes, they can make it just not as well as dendritic cells. And I will also say that there's a huge gaping piece of immunology that I'm leaving out in answering that question. Does that work? Okay. And so the next two slides are basically things I've already told you. So verbally, so I think I'm not going to show you those. But what I will just tell you is that if I want to turn on a T cell in a dish, I need to know this same information. And so here I might look at whether or not T cells are proliferating. I might give them some drugs that give them T cell receptor stimulation, like PMA or anti-CD3. And you can see they kind of like are okay. But if I add signal 2, if I add CD28, so PMA plus CD28 instead of just PMA alone, I get way better T cell activation because now I'm actually giving my T cell signal 1 and signal 2. Same thing. Here it's a signal 1. Here I give them signal 1 and signal 2. I actually get good T cell activation. So we have to use this information in a lot of our experiments. And it's a key thing to be remembering for a lot of our experiments. So there is another piece to the co-stimulation issue that we need to think about, which is that if I'm being fully transparent, there are tons of co-stimulatory molecules on T cells. So in this image, you can see MHC and signal 1, and you can so the yellowy cell is the T cell, the brownish cell is the antigen presenting cell, and you can see there are all sorts of different protein-protein interactions that could be there. Usually all of these are not happening at the same time. It's more like these are choices. Like some cells will have one kind, some cells will have another kind. But this is just kind of showing you all the different options. One really important one is CD28, but there are some other really important ones as well. And the one that I want to mention to you, well, I'm going to mention two of them, but the one that's particularly important for us here is called CTLA4. And so we're also going to look at CTLA4. I will also mention one called PD1, because it's kind of famous at this point. And what I want you to notice in the image on the left with all the different possible co-stimulatory molecules is that when we look at the T cell, some of those types of signals give you green plus signs. Other ones give you red minus signs. The idea is that some of these signals help turn the T cell on, while other signals turn the T cell off or help turn the T cell off. And these two that I want to mention to you, CTLA4 and PD1, are both inhibitory receptors. They both can turn the T cell off. And so here you can see, here you can see the details with CTLA4. And so if we have our naive T cell, which you can see right here, our naive T cell will have the T cell receptor binding to MHC. That T cell will also have CD28 binding to B7. We'll give this T cell signal 1 and signal 2, and we'll turn that T cell on. If you think about it, going way back to earlier today, we're probably also this cell is going to turn on new genes like IL-2, right? And like IL-2 receptor, so that it can start changing up its responses. Well, that cell is actually going to start making, the T cell is going to start making another new gene as well. And the new gene that this T cell is going to start making is this gene called CTLA4, which you can see here. you can also see this on this next slide. So here's my naive T cell. It's eventually going to get activated. It's going to start making things like the IL-2 receptor. It's also going to start making CTLA-4. Sort of as it gets activated, it's going to start making CTLA-4. CTLA-4 is really cool because CTLA-4 also binds B7, as does CD28. Binds the same thing that the signal 2 protein does. But CTLA-4 will provide a negative signal. It will stop the T cell from being activated. It will, in fact, turn the T cell off. We'll see some of these receptors more later on, but if you remember, I told you about ITAMs and activation. CTLA-4 has this thing called an ITIM, the immunotyrosine-based inhibition motif, because it's an off switch instead of an on switch. CTLA-4 binds to B7 better than CD28 does. This is a much stronger binding than the CD28 B7 binding. And so what happens is that when our naive cell becomes activated, the B set and starts to make CTLA-4, the CTLA-4 is going to stop binding to CD28. It's going to start binding to CTLA-4. It's like a competition. And CTLA-4 is the winner because there's better binding. So now you can see the B7 switched. It's not binding to CD28 anymore. Instead, it's binding to CTLA-4 because this binding is so much better. And the idea is as soon as you activate a T cell, that T cell starts making an off switch. So that T cell can be turned off. Because we don't want excess T cell activation. If we get excess T cell activation, we're going to end up with autoimmunity. And so as soon as we turn that T cell on, the T cell is also going to start putting an off switch on its surface so that it can be turned off later on. Yeah, Brooke.
Can you turn it back on?
Yes, you can turn it back on. Yeah, so this is not an anergic cell, it's just an off cell. What's sort of also particularly important here, this sort of gets to my Josh's question and makes me realize more about how that oversimplification. In the periphery, or whenever a T cell is not naive, the T cell only needs signal 1. So if we're not in the lymph node, we're probably not going to see a ton of B7 at this point. So we're not going to turn the T cell off. We're just going to let that T cell get signals, get turned on. It's just that later on, that T cell, if it sees B7, will get turned off. So if my T cell is in the lymph node, so my lymph node is from my sinuses, it's going to see B7. It's then going to move to my nose because it's been activated in my lymph node. Now it doesn't need B7 anymore. Now it just needs signal 1. It doesn't need signal 2. Well, it's in my nose. It's going to kill all of the microbial infected cells in my nose. And then maybe it might head back to the lymph node at some point. It's going to see B7 again and be like, okay, I'm done. I'm off. Does that make sense? Okay. And so here you can see that expression of CTLA-4 by activated T cells. As I mentioned, I did want to say some brief stuff about PD-1. If you look at the top here, you can see we've got a naive T cell. So let's say a T cell that's never been turned on before, or in Brooke's case, her question she just asked, a resting T cell, so one that got turned off before. It's going to use signal 1 and signal 2 from CD28. It's going to get turned on. It's going to eventually get CTLA-4 on its surface and get turned off. If that T cell is perhaps not a naive T cell, perhaps that T cell is an activated T cell in the periphery, it might use a different negative receptor, like PD-1. And so you can see there's this other receptor, PD-1, on the surface of the antigen experienced T cell that might help that antigen experienced T cell get turned off in peripheral tissues. And so you can see, I showed you that slide before that said there were so many co-stimulatory molecules, and there are so many co-stimulatory molecules. One of the big things that differs from them is if they're on, say, naive versus activated cells, where they're involved in turning cells off. And PD-1 can give that same kind of negative signal to an antigen experienced cell in the periphery. Brenna.
I don't know how to word this question.
Okay.
But I know you said in the periphery it doesn't need signal 2.
Mm-hmm.
But is B7 always going to be needed? So, like, is it just dropped? Like, how does it just drop it? Because you said it doesn't need it.
I mean, I know the answer. It's just that I don't know that I want to give you that much rabbit hole.
Basically, B7 doesn't always have to be made, or it is always made.
Dendritic cells are super good at making B7. Everybody else is meh. Okay? And so that first time when we're turning on our T cell in the secondary lymphoid organ, in the lymph node, it's a dendritic cell that's doing it, that makes super awesome B7. There might be a little around, but we kind of don't care about it in the other places. It's meh. Okay? One other thing that we've noticed about PD-1 is that if a T cell sees antigen for a long period of time, that T cell, so here's the T cell just seeing the antigen for a short period of time, maybe like you get the flu and you get over it. Here's a situation where maybe we see the antigen for a long period of time. That T cell is going to start making PD-1, and you can see it starts making like a lot of PD-1 over time. This process, you're going to understand this process pretty well. This process that happens if a T cell, so what would happen if, we talked about when the exam would be, right? We're going to have an exam, and you're going to have some stress of studying for the exam, right? And then you're going to not be studying for the exam. What would happen if I gave you an exam every day? How would you feel? Okay, all right. Let's go with the other answer that we heard from somebody else say. How would we feel if we had an exam every day? Stressed, right? How would you probably feel by the end of the semester? Well, or why? Dead in what way?
I would be
burned out. This process where a T cell sees so much antigen is actually called T cell exhaustion. You'd feel exhausted, right? And so it seems like T cells in particular make a lot of PD-1 when they're exhausted. And when T cells are making a lot of PD-1, they don't seem to function really well. It seems like they make a lot of PD-1 and get exhausted when there's chronic infection, like an infection that's happening for months and months and months, years and years and years. It also seems like they get, we sometimes will see some of these chronic issues in cancers, as well as some other situations where the T cells start making a bunch of PD-1 and they're just exhausted. The reason why I mention all of this is that it's like 5 years ago now. It feels like it just happened. There was a Nobel Prize given for the discovery of something called checkpoint inhibition. I remember when the checkpoint inhibition papers came out, where we realized that if we actually can inhibit CTLA4 or we inhibit PD-1 using an antibody, we can keep T cells from getting exhausted. We can keep T cells from getting turned off. And in some clinical situations, T cells getting turned off too early, or T cells getting exhausted, is the problem. If the T cells would just keep working, then we could then the disease would get cured. And so if we use something to block CTLA4 or PD-1, then we actually keep the T cells on, keep the T cells working, and can treat the disease. So this idea of checkpoint inhibition, either of blocking PD-1 or of blocking PD1, or sorry, CTLA4 or PD1 is being used for more and more and more disease states, particularly cancer treatments all the time. If any of you guys have heard of Keytruda, Opdivo, pembrolizumab, Pembro is sort of the short version. These are all checkpoint inhibition drugs, and they have really dramatically improved lifespan for a lot of patients. And they tie into what we see here with T cell activation. So, at this point, we've had our naive T cell. That naive T cell has gotten signal 1. It's also gotten signal 2. This actually shows it getting signal 1 and signal 2. It's made some cytokines. It's gotten those cytokines to feed back to itself. It's undergone some division. You have your nice swollen lymph node. But one other big thing has happened to that T cell. That T cell now has undergone a process of differentiation. And what that means is that this cell has now gained a function, or gained a function it didn't have before. Before, it was just a cell kind of hanging out, not really able to do a ton of stuff. But after it's gone through all of these steps, now the cell has an effector function. It has a function it can do in order to actually fight microbes. If that cell started out as a naive CD8 T cell, it will gain the effector function to kill target cells. It will become a cytotoxic T lymphocyte, or CTL. And you'll see that on some later slides. But the point I want you to think about here is that this original naive CD8 cell, this cell cannot kill other cells. This cell, when it first sees antigen, it's not going to kill this dendritic cell. It's going to get activated. And only after activation is this cell the killer. If the cell was a naive CD4 cell, after differentiation, that cell is going to make a lot of cytokines and become a helper T cell. This naive CD4 cell is not going to be making a ton of cytokines, but now that it goes through this activation process, it's going to be the helper and make a lot of cytokines. I'm going to say a little bit about the CTLs right now. We'll say more about the CTLs on Wednesday, and we'll also say all about the CD4s on Wednesday. But here you can kind of see this process. So originally, see, it's a dendritic cell, because dendritic cells are so good at turning on T cells. Here's my dendritic cell. It's turning on my naive T cell with signal 1 and signal 2. We're going to get some IL-2 production. This cell is going to divide a lot. Yay. And now, after this cell has divided a lot, this cell has gained its new function. And so now, this cell is going to be able to actually kill when it sees MHC. And you can also notice it doesn't even need signal 2 anymore. It just needs MHC. So this type of cell is known as a cytotoxic T lymphocyte, where this cell is going to be able to interact with those infected cells and kill them. And you can also see this process here. What I want you to notice about this process is that the CTL is specific. It is going to interact with one infected cell and kill it. It's not like it's going to destroy the whole tissue. It's going to pick an individual cell and be like, I'm going to kill this one cell. It's not going to damage like the whole lung or something. So it's a very specific interaction, but that cell does have the ability to detach and go kill another cell. And so here we actually see it killing multiple cell types. And so you can see how that recognition and killing is happening. I don't know if this is going to work, but I will try it. We're going to talk about the specific molecules that the cell uses to do that killing on Wednesday. Oh, I didn't know there was music. All right, so we can see some T cells hanging around. Yay, exciting T cells. It's because the one I want is like gone. The video I want. Ah, there we go. Here's our T cell. And now it's going to interact with an infected cell. And it's going to induce apoptosis of that cell. By secreting specific proteins that are going to lead to the death of that cell. And so I will find the good video of this to show you for Wednesday, and you'll get to see how this process really works.